I think we should start, sir. So, good morning to all of you. Today, again, we have with us Dr. Shogata Bhattacharya, sir, and he is delivering the lecture on this said topic. So, I kindly request to Dr. Shogata, sir, please share your slide and deliver the lecture.
Yes. No slide is open now to all of you.
Yes.
Yeah. Okay. Okay. Thank you, and very good morning to all of you. Welcome you again. Now today's topic is concept of bioinformatics, genomics, and proteomics. So at the initial time, I should say that you must have the knowledge of genomics. Okay, so I will start with genomics, proteomics, and finally I will come to bioinformatics, because bioinformatics is the application of genomics. So what is genomics? Genomics is nothing but an interdisciplinary field of biology which focuses on the structure, function, and the evolution of a species, the mapping, and the manipulation of the genome. So a genome of an organism is the complete set of DNA that contains all of its genes, and the term genomics was first time coined by Tom Roderick in 90s. But the advancement of the recent technologies like genome sequencing, the high throughput genome sequencing platforms, the genome sequencing projects are nowadays coming. So as you all know, the first human genome sequencing project was started 2001 and it was completed in 2003. So that time the revolution of the genomics happened in case of animal in terms of animal biotechnology. But after that, so many crops has been sequenced in agricultural perspective also, like Arabidopsis was sequenced, and a database has been created for TEAR, the Arabidopsis information resources. Okay, so there are after that in 2004 another crop, that is rice, was completely sequenced. Now 2018 the fine genome sequence of the wheat has also already came, and so many drop sequences are already available. So we will see the how how the field of genomics has been revolutionized. Okay, so the first eukaryotic chromosome, if I say if the question asked in exam that what was the first eukaryotic chromosome was sequenced, that was the Saccharomyces cerevisiae. Okay, East sequence was the first eukaryotic chromosome. But if they ask what was the first eukaryotic complete set of sequence or organelle was sequenced, that was human mitochondria only. That was 16 kb in length. Okay, so these kind of database questions can be asked in case of ICR exams like SRF. That you must remember what are the genome that was first sequenced, what was the first virus, what was the first bacteria, what was the first eukaryotic sequence, what was the first eukaryotic multicellular sequence, okay, the first organelle sequence. So this kind of data you have to remember. Now, the suffix om is used to address the object of study of such fields such as the genome, proteome, metabolome. The suffix om is used in molecular biology that is basically the totality of the short. Okay, so omics refers to the large, the comprehensive biological data set. Now, if I say about genome project, now first you must understand the what are the different different arena of genomics. Okay, so if we see the central dogma, the genomics, if we start from chromosome has a DNA, and DNA has different component. I will come. DNA has coding, non-coding, tandem repeats. Okay, then transposomal elements like line, sign, also called retrotransposomal element, and LTR, non-LTR. These so many things in genome, because the total chromosome, the only 1.5% to 2% are expressing in case of eukaryotes, and in case of prokaryotes 90% of are expressing. So must remember the only 1.5 to 2% of the chromosome is giving actual protein or a functional protein, and rest of the things are full of tandem repeats, non-coding elements, transposomes, etc. Okay, so in case of from DNA to it is translated transcribed to RNA, which is called transcription, and followed by translation, which is synthesizing protein. And then the protein has different function. It may act as an enzyme, it may act as a structural protein, and so on. So we have to study the metabolome also. So if we say what is functional genomics and what is structural genomics, now functional genomics only comprises of transcriptomics and proteomics. Okay, together we both call them as a functional genomics. Do not misunderstood this concept. The functional genomics comprises both, okay, transcriptomics and proteomics. Now, as I said, genomics is a field. It is a multidisciplinary field. Okay, it has Mendelian genetics, it has cytogenetics, molecular genetics, biochemistry, and so on. And it has so many parameters. That is, for structural genomics, we have to go for another approach. For functional genomics, we have different approach. Comparative genomics, this is a new concept, comparative genomics. For example, we do BLAST. BLAST is nothing but we're searching a query against a database. That means we're doing comparison. Then, and these are all the parts of bioinformatics. Bioinformatics, as the name suggests, it comprises of biology and computational biology. Okay, so if we put together, they form the bioinformatics, and then artificial intelligence, robotics, etc. are there. So together they form as a system biology. System biology is a new concept where the genomics, bioinformatics, computational biology together came into a picture. So if we have a target selection, then what to do? I have to sequence a genome. Okay, I have to sequence a genome. For that, we have different approaches, like we can go for whole genome shotgun approaches. We have to cut the chromosome into small pieces. We will, what I am telling, that was the way back system. Okay, so I have a chromosome, I have to cut those chromosome into fragments, put it into a cloning vector, and then we will again subclone it, because at that time the YAC or BAC was used, but YAC and BAC size is almost 300 kb. That was the insertion size for BAC, and for YAC it was 1400 to 1500 kb size. So that time, that much sequence amplification of that much length, or sequencing of that much length of a chromosome fragment was not possible. So we have to subclone it into a fudge-meat or cosmid vector, make it small, and then we will go for sequencing like Sanger sequencing. That was the primitive form of sequencing that I am telling about, the classical Sanger sequencing, conventional one. Okay, now that was called hierarchical shotgun sequencing, and then go for high throughput. Nowadays, a high throughput sequencing technologies are available. So we will prepare a draft sequence, then we will complete the sequence, that is called finished sequence or fine sequence, and we will archive that sequence in our database. So after that, we have to go for genome annotation. Okay, so after sequencing, we will do annotation. Annotation means assigning a function to a gene. Remember this terminology, assigning a function to a gene. That means we have a sequence, then we have to identify a function for that particular sequence. Okay, so that can be done in different forms, like we have to give them, put that particular sequence. If it is a protein coding, then it must have some role. In what pathway it is involved, and different kind of comparative analysis we can do. Okay, now in terms, since I told the genomics has role in different arena, like in molecular medicine, identification of a component of a particular medicines, or how design a new medicines. Okay, and what are the expression pattern, gene therapy? There are different function in molecular medicine. Then we have also genomics has also role in developing targeted drugs. That's why that is called pharmacogenomics. That is another area of genomics. Then toxicogenomics. Okay, elucidation of a gene that which has that are affected by various chemicals. Okay, so what are the what are the role of that particular toxin? Just yesterday I was telling that for transgenic crop also we have to go for a toxicology study, that whether it has oral toxicity or not, what are the similarity of that particular toxin with the other known toxins. Okay, so there are genomics is a vast area. So today we will discuss mainly focus on agriculture. So as I see in that picture, that in terms of crop improvement, how genomics is playing a role. Okay, so in terms of genomics and transcriptomics, as we see, we have sequencing technology. So after getting that sequence, we can go for RNA-seq, we can go for microarray, and this kind of technology helps in identification of a particular gene or QTL or any marker, or we can identify a particular pathway. Then definitely we will go for a improvement of that particular aspect. So and if I come, if we tell about bioinformatics, I will cover these things, that is database. What are the different database? What are the sequencing platforms nowadays available? How assembly we do? What are the assembler, the tool available? And how genome annotation is done? That means how we assign a function to a gene, and the GWAS, that is genome wide association study. And we will discuss this thing one by one. Now coming to database. Okay, so database in bioinformatics, it is bioinformatics, as I told, it is a not only in case of bioinformatics, but also any kind of area or discipline that deals with the intensive research. Okay, so database is needed. So for database, in case of bioinformatics, we have two kinds of database. One is primary database and secondary database. So what is primary database? Primary database are elementary derived data. It encompasses the elementary data. That means nucleotide sequence, protein sequence, macromolecular, in any kind of sequence. So this kind of data we put into primary database. Okay, you have a nucleotide sequence, you have cloned a gene, you have sequenced a gene, you put it to NCBI. This is primary database. So NCBI, Gene Bank, DDBJ, these kind of database are primary database. And coming to secondary database, which is a curated one. Okay, so after analyzing the result of a primary database, if you are doing some curation of the database and you have some addition of the elementary analysis of the primary data, then we will put it into secondary database. For example, InterProScan, UniProt, okay, Ensemble, these kind of database comes under secondary database. So this is clear. The primary database, NCBI, Gene Bank, sometimes question asks which of the following is a primary database. Okay, so remember NCBI, Gene Bank, DDBJ, these are primary database. And if we curate some data which was taken from the primary database, suppose one example, you have taken a data from NCBI and you have done some analysis and you put it in a database, that will be a secondary database. Okay. Now coming to BLAST, there are different kinds of BLAST tools are available. For example, BLAST-N, BLAST-P, BLAST-X, T-BLAST-N, T-BLAST-X. Now in coming days, there are SciBLAST, MEGABLAST, these kind of concepts are there. So you must remember these things, because question asked in every year from the BLAST types. Okay, what are the different types of BLAST? And question will be asked that this is the query and this is the database, which kind of BLAST program we will run. So the question will ask like this. So remember, I will not going through this. So you have to remember, no other way, and try to remember these things using an example. For example, you go to NCBI site, you search BLAST, and these options will be available. Along with, I said, SciBLAST, PyBLAST, then MEGABLAST, these kind of options are also there. Nowadays, IgBLAST, immunoglobulin protein search, CDBLAST, okay, conserved domain search, these kind of BLAST are also available. So you have to remember all those things, because question will definitely come from this area. Okay. Now, if we come to genome mapping, the genome mapping is a old concept. This is not a new concept. What we do recently, that we have to find the mapping what we get from the raw data. For example, I have a draft data. For example, in PGNP, there is a draft sequence available from the variety that was sequenced was ASA. So the draft sequence is available, but fine sequence is not available till now. So for that, we are improving more and more day by day. Fine mapping is the area where currently bioinformatics people are doing a lot of research work. But here I am telling you what are the basics of genome mapping, why we need mapping. So genome mapping is basically different from gene mapping. Okay, gene mapping means you have allocation of a particular locus. You have to allocate a particular locus to a relative position in the chromosome. Okay, so genetic mapping is something that is species specific, that has to identify what are the markers, what are the genes, and what are the distance between the marker and the genes. These kind of things are called genetic mapping. So genetic mapping is a relative position of a gene from its marker. So generally it measures as a centimorgan, and the centimorgan value can vary from crop to crop. In Arabidopsis, 1 centimorgan is something. In rice, 1 centimorgan is something. So that is a relative term. Okay. Now coming to cytogenetic map. So cytogenetic map is nothing but a systematic representation of the chromosome that indicates the arrangement of the individual genes. Okay. So cytological maps of organism is basically a map that the relative position of the gene. We represent the relative position of the gene, and no marker is available. For example, a chromosome has P arm and Q arm, the smaller and larger. So we will give a different color or pictorial depictions. That is cytogenetic map. Okay, and another is physical map. Physical map is the ultimate map after sequencing. Okay, so physical map is a technique in the molecular biology to find the exact order and what are the exact physical distance. I am telling the word exact distance and exact order of the DNA base pair by identifying different markers. Different molecular markers are available, as you have heard of, that RFLP, AFLP, then SNP markers, SSR markers. Okay, there are so many lot of molecular markers that we can use for identification for getting a physical map. So in case of physical map, FISH is another technology. Yesterday I discussed. Then sequence, tag site, EST. This EST is a single pass cDNA sequence. So this kind of sequence information is another also can be used as a marker for constructing physical map. Okay, now so you have a reference genome. If you have a reference genome, you can go for reference assembly. If you don't have a reference genome, then you have to go for de novo assembly. So I will come into that. So before that, the bridge is the DNA sequencing technology. Okay, so you have isolated a chromosome, you have fragmented those chromosomes. Now you have to sequence those chromosomes. So in recent year, there's a first generation, second generation, third generation, and so many development is going on with time. Okay, so and the development is not basically based on the sequencing, the development in the aspect of the accuracy of the sequencing and the read length. That is the major two constraints of the sequencing platform, that you have to get a good read length so that the number of read will be less, and another parameter is the accuracy. You have to get a 99.9% accurate result, so that the draft sequence, the labor we give to get a fine sequence from a draft sequence will be drastically reduced, because fine mapping is very difficult. Okay, so the first sequencing technology, that is Sanger sequencing, and this is still a very good platform because nowadays we have automated Sanger sequencing. Okay, so what we do in case of Sanger sequencing basically also called a chain termination sequencing technology. Because here we use a DNTP. Just remember this picture. We normally we amplify a DNA through a DNA polymerase using DNTPs. Okay, so it will add one by one to the newly synthesized chain. But if we add DNTPs, because the 3' OH is replaced by H. Okay, remember this thing. What is the difference between DNTP and DNTP? Question may ask from this also. So here OH is replaced by H. So 3' N is not available. So as we know, the phosphodiester bond formation, 3' OH and 5' phosphate is responsible. Okay, if we replace this OH with H, then it will not be able to amplify the DNA now after. So this is the basic simple technology. So what Sanger did. Sanger used different labeled DNTPs. Okay, labeled DDNTPs. For example, here DDTTP, CTP, ATP, GTP. So all the dideoxy NTPs were used with fluorescent label. Okay, so if we amplify those sequences, so what are the steps? The double-stranded DNA is denatured and make it into two-stranded. Now it is single-stranded. The primer that correspond to one end of the sequence is attached, and the 4 DDNTPs was labeled, and the DNA synthesis reaction initiates, and the chain extends until the termination happens. Okay. So whenever it will get DDNTP, the chain will terminate. The resulting DNA fragment are denatured in a denatured gel electrophoresis or capillary gel electrophoresis, and the sequence will be scored. Here, after if we score the sequence using that chromatogram, here we can see that this is the different fragments we get. Okay. Now, how to read or analyze this sequence? Question asked from that part only. Every year, question is coming from that part only. That how, if you get at this, suppose they will give you this this kind of sequence, okay, 3' to 5' or 5' to 3', anything. So you have to answer the questions based on 5' to 3' only. Just remember this thing, what I am telling. Always answer the questions from 5' to 3'. And so 5' to 3' will be the complementarity of this sequence. So how we read? We read the sequence from bottom to top. Okay. So that this has come at the base level. That means, so we will count these sequences first. So first, second, third, fourth, fifth. So from bottom to top, we will count it, and then we will convert it as 5' to 3', because sequencing happens always from 5' to 3'. And what you are getting, that is the complementary strand. What you are getting, the complementary strand. This was the complementary strand, newly synthesized DNA. But question will be asked that what is the template strand sequence? So the answer will be this. So always remember 2 things. You have to count from bottom to top and convert it into 5' to 3'. The answer is correct now. Now, the second technology is pyrosequencing, also called Roche 454 FLX system. Okay. So pyrosequencing for any kind of, first let me tell, listen that for any kind of DNA sequencing technology, what we use nowadays always uses a library preparation. Okay, the library preparation is the most difficult part. Sequencing will do the machine itself. Okay, so the template, what we do, the template can be from any part of cDNA, single mRNA, based on your desire, what you want to do for what you are want to do sequencing. Okay, then after that we have to digest it either by nebulization, by sonication, or enzymatic reaction. So make it to small fragments of suitable size, and then adapter will be ligated. Adapter will be ligated at both the end, 5', 3' end, and then we'll go for a amplification method, that is PCR. Okay, so and how we do PCR? For PCR, if there is a microflow cell, and in the cell there is a small beads. Okay, and now the fragments are attached to a bead. So each single bead has a single kind of fragment. Okay, that means each bead represent a particular fragment, and then we will give PCR enzymes and reagents, and then the droplet, that is called now emulsion. It is an emulsion of oil, the fragment, okay, and all the PCR components. Now this is called emulsion, and the PCR will take place in a microreactor chamber. So this is the this is common for all the next generation sequencing platforms. Okay, now for pyrosequencing, what do we do? That, as I said, each well contain only one bead, and one bead has one kind of fragments. So what we'll do, the principle is that is based on the Luciferase based detection technology. You have to remember the principle only for each sequencing. Do not have to remember all the steps and all the small things, but so remember what is the principle of this technology, that whenever a DNA polymerase adding a particular nucleotide, it will release a fluorescence. Okay, the fluorescence is basically depends on the phosphate we use. For example, it has let me explain the steps that you are using. Whenever a PPI, whenever you are adding a DNTPs, it will release a PPI, pyrophosphate, and the pyrophosphate, we're adding enzyme ATP sulfurylase, so it will convert to ATP. Okay, now the ATP will react with luciferin, so it will give a oxyluciferin, that is a light detection. Okay, and then so apirase enzyme is added. Okay, now the DNTPs will give rise into the phosphate. And remember, apirase was extracted from potato. Okay, from potato, apirase is generally commercially extracted. So a pyrase reaction that degrades the NTP converted into PI. So you will get a visible light also, you will get a PI also, and the PI, the potentiality of the PI varies with each nucleotide. So ATP has different, CTP has different, GTP has different. So accordingly, the detector will capture the which kind of base has been added. The beauty of this technology is it has a larger read length. It can read up to 1000, more than 750 base pair, and it has 99.9% accuracy. So this is the beauty of this technology. And nowadays everything is automated. So detection, you will get a chromatogram and you have to analyze the data only. So this is better, this is more perfect than Sanger sequencing, but Sanger sequencing, automated Sanger sequencing is still now very useful. Now coming to Illumina Solexa, this is the most widely used in this technology till date. Remember, this is the most widely used technology till date, though it has a small read size. The small read size is only 150 to 200 base pair, but still now very useful because of its accuracy and speed. Because nowadays we need a huge amount of data in small time. So that's why this technology is leading. Okay, there are so many platforms. You can use Illumina Genome Analyzer platform, HiSeq platform. Okay, so in this technology, two different adapters are ligated to the 5' and 3' end of the any kind of fragment you are using. So here we can see 3' and 5' fragment, the adapter are ligated, and these fragments are attached to a substrate that is called a flow cell. Okay, so remember the name flow cell. So, they are ligated to a flow cell, and a solid phase PCR, also called fold back PCR or bridge PCR, is performed, because in whatever adapter we have added in the flow cell, there will be the complementary adapter, so it will bind. And another point, another flow cell, there is a for example, this is adapter 1, this is adapter 2. So in adapter 1, complementarity will be one flow cell, and adapter 2 complementarity will be another flow cell. So it will form a bridge, okay, or you can say a fold back. That's why it is called fold back PCR or bridge PCR. Sometimes questions are bridge PCR is the technology for which in this platform. Okay, Illumina. For Illumina, bridge PCR or fold back PCR is there. So the 4 fluorophores that is labeled with DNTPs, and it will be detected. And as I told, this is almost 98.9% accurate, and it can perform 400 GB of sequence data within 7 to 8 days. Can you imagine 400 GB of data you will get in only 7 to 8 days? So that's why this is the beauty of this technology. But as I said, the read length is very small, that is only 50 to 150 base pair. Now coming to PacBio, that is also called smart technology.
Did you know, Sheikh?
So smart technology, that is single. What is the meaning of smart? Smart means single molecule real-time sequencing. Okay, as the name suggests, single molecule. That means if we have a single-stranded DNA or a RNA or cDNA, then you can go for PacBio or smart sequencing. And this technology was developed by Nanofluidics. There is another company, Nanofluidics, and was commercialized by Pacific Bioscience. That's why the instrument name PacBio. That was the company of USA. And in this technology, template is prepared, okay, through ligation single stranded hairpin structure. So a adapter is added with that is a hair that has a single stranded hairpin structure at the end of the cDNA. For example, you are taking a single stranded cDNA. Now, now the shape of the product, the complete product, after adding the hairpin adapter, will be like a bell shape. That is called smart bell. Now the single molecule DNA polymerase are immobilized at the bottom of the G-politer size well. Now this is called G-politer. Okay, this is a G-politer. That means the size of the well is G-politer size. And here a DNA polymerase is immobilized. The enzyme is mobilized. Enzyme mobilization, there are so many techniques. There is cross-linking. Okay, so here it has been immobilized. Now you will pass your single molecule with that G-politer well. Okay, and fluorescent level detection will be detected by the displacing the polymer. Whenever a passing your molecule and it is starting synthesizing the adding the DNTPs or starting synthesizing the new strand, so you will get a detection and at the growing 3' end. Okay, so this smart technology is very specific. You can say this is almost 99.9% specific, but the problem is the sequence read length is very small. Read length, I am telling this word repeatedly. Read length means at a single time, a single pass, how much base has been added. So the read length is very small. That is only 50. Sorry. What I was saying, that the beauty of this technology is it has a higher read length, that is 50 kb, but the accuracy is somehow 98% or 97%. This is the problem. Okay, whenever you are getting high read length, the accuracy will be less. Okay, for example, solid, I have not mentioned here, solid, this oligonucleotide based method, that is 100% accurate, but the read length is only 50 base pair. See, and now in case of PacBio, you are having 97% or 98% accuracy. That accuracy you are compromising, but you will getting a read length of 50 kb. That is huge. Okay, and for 1 GB of data, it takes only 4 hours of run. Okay, so this is the problem and beauty of every NGS technology. Now, the recent technology, that is Oxford Nanopore. Oxford Nanopore is an advanced sequencing technology, and it uses a flow cell. The flow cell composed of hundreds of microwell, and just like Illumina. So it has a flow cell that has hundreds of microwell in that flow cell, and sequencing is achieved simply by precising measure of the change of the current. So whenever there will be a sequencing, there will be change in the current. So automatically it will detect that current, and that results in the incorporation of the bases, which indicate that the base has been incorporated to the newly synthesized strand, and there is a molecular motor protein. That protein helps in detection. Okay, so here we can see there is a nanopore, and cyclodextrin is used to capture the molecule, and whenever there is a entry, but what is the difference? Actually, the difference is here we are not synthesizing. Actually, we are degrading. You must remember this terminology. Oxford Nanopore is not sequencing by synthesis. This is sequencing by degradation of the DNA. Because here we are using exonuclease. So whenever a single molecule passes through the nanopore, the exonuclease present in that nanopore will cut one base at a time, and that base releases a current, change in the current, and that will be detected by the motor protein. So this is the principle of the Oxford Nanopore. So sequencing by degradation. Now, coming to genome assembly. Genome assembly, why we need assembly? Actually, the assembly is simply the after sequencing, what are the products we have? That means the fragment of different chromosomes. Then we have to put them back together. So through bioinformatics, the sequence assembly we can do through alignment and merging of the fragments. Okay, and to form or reconstruct the original sequence. Clear? So the need of this genome assembly is because we do not have any sequencing technology platform, as I told now, that we don't have any sequencing technology platform that can sequence a read length of more than 50 kb or 300 kb. So we have to make them very small fragments. So these small fragments, as we said, that in case of PacBio, we are getting only 50 kb maximum. But in case of Illumina, that is most widely used, what we are getting? We are getting only 50 to 150 base pair. So this is the read length. Now we have to assemble those reads into a single, to make a single chromosome. So that's why assembly is needed. Now for assembly, there are two kind of assembly tool. One is comparative assembly and is de novo assembly. Comparative assembly means when you have a reference genome. For example, rice, oryza, sativa, you have complete sequence, for example. But you want to sequence now oryza nivara or oryza japonica. So in that case, you can use rice oryza sativa sequence as a reference sequence because you are sequencing the same species. So you can get a help of a reference sequence. But in case of a you have to identify a sequence of a new crop. For example, the oryza sativa, that first time was sequenced, that time it has no reference sequence. So only de novo assembly is the options. Okay, clear? So in case of comparative genome assembly, another terminology I am saying here, one is scaffold and what is contig and what is coverage. This kind of concept you must have. Scaffold, first, what is contig? When you are joining few reads into one stretch, okay, based on their order of the bases and with high confidence level, not like that you have somehow put those reads. This is not with high confidence level with using the different libraries, made pair libraries, parent sequence. So there are different approaches. So you are making the reads together at a stretch. This is called contig. That means contiguous sequence, contiguous length of genomic sequence. Okay. Now, what is scaffold? When so many contigs you are putting together, but there may be few gaps between two contigs because so many problems are there. There will be repeating elements, there will be heterozygosity, there will be polymorphism, there will be SNPs. So there will be some mismatch with the reference genome. Okay. So this, whenever you are putting more than one contigs together, now the stretch is called scaffold, but scaffold must have gaps. The gaps may be sequence gaps. Okay. And so coming to comparative genome assembly and coverage. What is coverage? Coverage, sometimes we say 10x coverage, 100x coverage. The coverage means the number of read into average read length. Okay, number of read. For example, you are getting 10 reads and the read length, average read length is suppose 7 or 20. So 20 into 10, number of reads into average length and total genome size. Okay, तो एवरेज रीड एंड एवरेज लेंथ को मल्टीप्लाई करके टोटल जीनोम साइज को अगर डिवाइड कर देंगे उससे, तो हमें जो मिलेगा दैट इज कवरेज. Generally, more 100x coverage, 1000x coverage is very much required for new sequence information. Okay, for that time you will have high confidence level. But in general, if we are doing resequencing of a particular gene or a particular genome, then 10x coverage is sufficient. Okay, but for fresh sequencing, 100x, 1000x coverage is required. So coming to comparative assembly. Comparative assembly is basically the alignment with the help of a reference genome. As I said, there are few steps. One is read alignment. Read alignment means each shotgun read, whatever read you have, that have to be aligned with the reference genome using any kind of computational tool. Generally, MAMR is a good software. And then repetitive sequence and polymorphism between the target and the reference genome. Okay, may be aligned due to non-continuous fashion, because I said that there will be a problem because of repeating sequence and because of polymorphism. So the longest increasing subsequent algorithm, that is called LIS algorithm. Remember, LIS algorithm is a part of read alignment of the comparative assembly. That LIS algorithm, that which has to generate a long chain that a mutually consistent match between the each read and the reference. As a mutually consisting match, whatever. So if you have small 10 read, but one read that is matching with the rest of the nine reads, and which is more reliable. What I mean is that longest read is more reliable than the small read when you are doing comparison with the reference sequences. So this is called LIS, longest increasing subsequent algorithm. Okay. Now the repeat resolution. Repeat resolution, what? The first, it is checked that the pair in sequence is uniquely anchored. There are two types of sequencing you can go for. One is single read and a pair read. Single read means you are sequencing from a one side, either 5' to 3' or 3' to 5'. And for pair end, we are doing both to increase the confidence level. If we can cross check the 5' to 3' and 3' to 5' sequence are doing similar, then that is more reliable. So the first step is check the pair end sequence are uniquely anchored with the genome. They are joining perfectly and satisfy that that they can impose in the mate pair information. And the second thing is that both the pairs are non-ambiguously placed. Okay, non-ambiguously placed. They satisfy the mate pair constants and distance and orientation. So based on their distance and based on their orientation. Sometimes direction is the problem. If we go for single end sequencing, for example, you have 3 reads, 1 by 1, and one reads at 5' to 3', another reads 3' to 5', and they somehow are giving a similarity with the reference genome, and there will be a blunder in that sequencing technology, in that assembly. Okay, so you have to go for a repeat resolution. Okay, to remove the repeats. So repeats are the major problems, and when the first 2 steps satisfied, now that can leave us the more than one placement of a pair. Then one possible placement may be that mate pair constants and choosing and random. Okay, so mate pair library preparation is very much required for repeat resolution. And then we come to layout refinement. Layout refinement is nothing but the another problem is indel. Okay, indel means insertion division. Between the target and the reference genome, there may be several indels in the read. So they may be somehow showing perfect homology with the reference, or maybe partially homology. So indel information can be reconstructed by traditional de novo assembly. There is no option. If there is an indel in the read length, then we can't go for comparative assembly. We have to go for de novo assembly. Okay. And then consensus generation. Consensus generation means for each group of overlapping read, now it is refined. That means it has no indel, it has no repeat, which is creating problem. Now we can go for multiple alignment using different computational tools. Okay, to generate a consensus sequence, there are different alignment tools, so you can go for that and make it consensus sequence. That is contigs. Make a contigs. So multiple alignment helps in forming contigs. Now finally we have to go for scaffolding. Scaffolding means based on the relative order and the direction of the contigs, we'll structure them in a linear way, and this contiguous region is now forming a scaffold. Now this scaffolding, whenever we complete scaffolding, now this is called pseudomolecule. Pseudomolecule means chromosome. Basically, a chromosome in genomics terminology is called pseudomolecule until and unless you have completed the fine sequencing, because there are so many gaps. You cannot say it as a chromosome. Okay, this is pseudomolecule. When you are removing the gaps after fine mapping, then now you can call it as a chromosome. So this is the basic terminologies and basic concepts of comparative assembly. And in case of de novo assembly, we have nothing, because we have no reference sequence to compare. So how we will do? We can go for OLC approach, that is called overlaid layout consensus approach. That means we have to arrange the reads one by one and see whether there is an overlap at the 5', 3' region. Is there any overlap? That means we can go for OLC approach. Okay, so finding the overlap, and then we will go for layout, and then a consensus will be formed. But the major drawback is whenever you are dealing with a large genome size. Okay, for example, you are dealing with onion genome size. Okay, and a wheat genome size is a very big genome. So in that case, OLC approach is not that much helpful, because there will be so many reads, and that will be very hectic, and there will be a chance of a false negative also. So for large genome, it is a very drawback of using this technology. And using OLC approach, what we will generally find, the minimum length of the overlap length. What should be the overlap length, minimum length? That parameter we have to set using whatever technology, whatever assembler we are using. For example, FRAP, Nublair, Mira, Ray, Abyss. There are so many platforms or assembler are there for de novo assembly, and Abyss and Mira, they are very, very popular platform. But we have to set the parameter, the minimum length of the overlap, the minimum similarity of the overlap, and the maximum length of the overhang which is not matching with the other reads. So what are the maximum length we can keep? The small length we have to keep, and the maximum number of local error we can consider. And the error may be due to sequencing error. Error may be due to the quality problem of the sequencing. Okay, these kind of things are there. So for small genome, OLC approach is very good. Okay, remember, for small genome size, OLC approach is very useful. Another method is de Bruijn graph method or KMR method, KMR graph method. De Bruijn graph method, generally KMR is any possible sequences of length k from a read obtained through DNA sequencing. Any possible sequences of length k. If you are getting a read length of k, then we can go for KMR approach, and you have to set that k value. Generally, k value 10 or k value 100. In between 10 and 100, we can set any value. And so, what is the possibility of getting a number of reads with that particular k value? Okay. So I will not going through this because this is core bioinformatics and so many statistics are involved. So just remember what are the name of the approaches and when we can use what kind of approaches. As I said, OLC approach can be used only for small genomes, and de Bruijn graph method can be used for large genome also. But you must remember what are the problems of assembly. The question can be asked from this region. As I said, the coverage. Coverage means how many times you are amplifying a particular read. As I told, coverage formula. So the length of the, as I told, the length and the number of read divided by total genome size. This is the formula of coverage. And problem is repeat, because repeat number can vary. Okay, while doing sequencing, if you are getting multiple times A or multiple times T, G, that times there may be a sequencing error. Sometimes while you are analyzing, maybe 2 or 3 G may be missing or may be added. So repeat is the problem. Then sequencing error may be a problem, because this is the DNA polymerase what you are using, the specificity of that enzyme. So these kind of things leading with that sequencing error. So this can be a problem. Polymorphism. Okay, polymorphism is another problem. SNPs are there, indel is there, and then heterozygosity. For example, in case of mango is a heterozygous crop. So you must have heard that the terminology that heterozygosity is a major problem in case of sequencing. Okay, so there is a solution for these problems also, that we can change the KMR value. We can set a neighbor KMR value. So these are core statistics. I'm not going into it. And then what are the assembler? As I told, for OLC approach, there are these assembler. Okay, for de Bruijn graph, you can go for Abbey's. Okay, so these all the assembler are using as a in case of de novo assembly. And if we go for if we go for comparative genome assembly, there are also different kinds of star. Okay, FRAP. These are also useful. For example, Trinity is highly useful in case of comparative genome assembly in transcriptomics. Okay, in transcriptomics, Trinity is very much used. For example, RNA-seq. Okay, these kind of things we can done with Trinity. So these are different assembler. Try to remember a few names, the popular names, and also remember in which category it belongs to. Then coming to genome annotation, as I told, annotation means assigning a function to a gene. Okay, so I have to identify the location of the gene and location of the gene and what the gene does. So there will be structural annotation and functional annotation. Basically, as I told, the structural annotation means what is the structure of the gene, what are the ORF, where it locates. Okay, what are the gene construct, what are the 5' element, what are the 3' element, what are the promoter, what are the regulatory motive, other regulatory motives, whether it is controlled by enhancer, whether any insulator is there or not. Okay, so this kind of information we can search as structural annotation. And if we go for functional annotation, that means what is the function? It may have function in any biochemical reaction. It may act as enzyme. It may act as a structural protein. Okay, so this kind of information we have to annotate. Then we will go for functional annotation. So there are 3 approaches. One is ab initio and hybrid and homology. Okay, what is homology? Homology means look for something that looks like one gene. Okay, homology means again comparative. You are looking for something that is look like one gene. Ab initio means you are looking for something that look like all the genes. So when you are searching something across all the species, then you will that is ab initio. When you are searching with a particular perspective, that is homology. you are searching homology. Homology can be orthology or paralogy, and hybrid if you are using both the approaches. For example, totally unknown protein, we have nothing to predict, then we will go for hybrid annotation. So this is three terminology only, and there are different annotation tools. Okay, so common assumption of a gene finding, you have to find a function of a gene and what is the structure of a gene. So common assumption is there is no overlap, there is no nested gene. Okay, there is no frame shifting, no sequencing error. Whatever information you get from sequencing platform, that means you have got a draft sequence and you want to find the function of that particular genes that are present in the particular chromosome, then we assume that there is no overlap. Okay, that means one gene is not overlapped with another. There is no nested. That means there is it is not the condition that in between a gene there is another gene has been inserted. Okay, there is nested condition, or no frame shifting of the sequence. There is no split start codon should not be splitted. Okay, that means in between start codon and stop codon there should not be any indel mutations. Okay, no alternate splicing is there, so no selenocysteine codon is there. That is ambiguousness is not there. So this is the assumption while we're doing annotation tools. This is not 100% perfect. So after annotation also we have to do functional characterization. That we do in case of transgenic research only. As I told yesterday, to identify a function of a gene. Function of a gene can be identified through bioinformatics only, through annotation tool only, but that is only prediction. That is not 100% accurate. So we have to go for transgenic research, or we have to give a mutation and functional characterize. We have to change, see the change of the phenotype. Okay, so these are the things. So as we can see in this chart that if we get to find the ORF, there are glimmer. Okay, fragment scan, metagen scan, there are so many tools. If we find a repeat, okay, so repeatory, there is another tool. Then if we annotate means give a function. After finding these kind of things, we can go for blast. If we're doing homology searching, okay, then we can go for p hammer. Okay, then so many tools are there. So metafiler is another excellent tool. So these are tools annotation for annotation. Okay, and I must suggest that few online cloud-based software is there, so you can register, you can use, for example, Galaxy is a cloud-based online tool that you can use for genome annotation. Okay, then Blast2Go is another tool. Okay, Blast2Gene Ontology, there's a tool Blast2Go, that's a paid software, but you can use it for any reference, using any reference example data to see how genome annotation is happening, what are the different parameters we have to set, just to remember these kind of things. Okay, and also remember few tools, name of the tools and what algorithm they are doing. Okay, now coming to Snp genotyping, as I told as I told yesterday, the Snp is the most polymorphic, most abundant marker. Most polymorphic marker is SSR, but most abundant marker in the genome is Snp. Okay, remember, Snp is the most abundant marker because you will get one Snp in 1000 base pair. Okay, or maybe less than 1000 base pair, you will get one Snp. So Snp is mostly diverse and most variant genotyping method to identify the variation of the two species or two individual. So identify the Snp genotyping is very much required. So in recent years, the Snp markers is the technology of choice for any kind of plant breeding program or plant genetic research. Okay, so available Snp discovery in the complex genome is required a efficient technology. Okay, because you have a genome size of 3.2 kb, 1.2 kb, okay, 1.2 GB, 3.2 GB, sometimes 15 GB, 16 GB. So identify the Snp in this large genome is very tedious. So multiplexing of the genotyping is required, and a high throughput, flexible, advanced technology is required. So how to identify that SNPs? There are a few technology I have given here. This is very simple. One is you can go for allele specific PCR. Okay, so you have if there is a mismatch in this technology, allele specific PCR means you are amplifying a particular fragment which is consisting of a mismatch, then it will give a signal. Okay, simply identify, remember the name, and if there is a mismatch, you will get a signal. So allele specific PCR is there. You can go for MALDI-TOF. MALDI means metric assisted laser desorption ionization time of flight. I will come this technology later. So in case of using MALDI-TOF also, this is a mass spectrometry. Nothing else is mass spectrometry. So you will get a peak that's showing that there is a difference in those peaks alignment, and definitely there is a SNP. Okay, using Tachman probe, as I discussed yesterday while discussing the real-time PCR probe mechanism, there is a Tachman probe. So Tachman probe is also useful because there is a reporter and quencher. Whenever there is a mismatch, there will be a cut. Okay, so you will get a signal using FRAP, fluorescence resonance energy transfer. So Tachman probe also can be used for detection of SNP. Okay, there are several methods. I have not mentioned everything because you have to remember the name only: allele specific PCR, MALDI-TOF, Tachman probe, invader method. Okay, then single standard chain electrophoresis. Okay, SSCP. So this kind of technology also very useful for SNP genotyping. Now, what is the use of the SNP genotyping? Because SNP genotyping, why you are doing? You are getting a marker information that is very robust. That is very advanced. Okay, now all the SNPs are not functional. Okay, so we have to identify few functional. So what is the application of SNP? SNP, we can go for GBS and we can go for GWAS. Okay, this is the two major classical examples I have given. One is GBS, genotyping by sequencing. Why we do GBS? Because we not all time needed the whole genome sequence. For example, for a particular character, for example, you are dealing with a branching pattern of a crop. Okay, you have to identify the why the branching pattern is varying in a particular population. So we have to sequence that branching because that crop is already annotated. That means we have the information of the gene responsible for the branching. So you have to sequence those genes only for all the population, all the individual of the population, and we have to compare. Okay, for example, you have 10 plants of a population, 10 plants and varying branching pattern. You amplify those 10 genes, those branching genes from those all 10 individual, and then go for alignment. Whenever there is a mismatch, you will get a SNP. That SNP is functional SNP because you are comparing of the same species of a population, so that you can get information that which portion of the gene is actually responsible for variation. Okay, so that's why we go for GBS. So GBS is a single nucleotide polymorphism detection. This is a method to discover SNP in order to perform the genotyping study. Okay, so for example, GWAS. I will come in the next slide. What is GWAS? Genome-wide association study. So what GBS uses? GBS uses restriction enzyme to reduce the genome complexity. As I told, the genome size is very big, so we have to reduce the genome complexity or genomic size. So we will go for restriction digestion, and after digestion, PCR is performed. Okay, and increase the fragment of the pools and and make a pool. Okay, after restriction digestion and PCR, we will go for normalization. Okay, after normalization, we will pool the samples of the older individuals, and then PCR cleaning and library analysis will be done after, and then we will go for sequencing. So this is a multiplex technology. This is a robust, simple, and very useful and inexpensive method for plant breeding. Inexpensive. Why I am saying inexpensive? Because we don't need to sequence the whole genome. We will reduce the sequence. We will just fragment those sequence, and we will amplify our desired product, and then we will go for sequencing. And whatever sequencing information we go for FASTQ. Okay. Just one thing I want to tell, that whenever you have a raw data sequence readout chip, okay, SRA data you will get from NCBI. For example, that is a sequencing data, just raw data. What we have to do with that? We cannot put it into the assembler for assembly, or we cannot put it into the annotation. What we have to done? We have to go for a quality check. FASTQ is the very useful technology. Okay, FASTQ, quality control. So first you have to convert the SRA data into FASTA data. Okay, FASTA format. FASTA is the format. Then FASTA format data will be put into the FASTQ software or FASTQ tool, so that you will get the quality of the data. What is the GC content? Okay, what is the read length? So quality you will get. If the quality is very low, if the GC content it is less than 40%, if the so many read length is not well or normalized, then you can simply say that this data is not good, you have to sequence again. Okay, the raw data is not good. But if you get good quality data in the first queue, then yes, you can confirm that we can go ahead. Then we will go for trigonometric. Trigonometric means we have to trim the data. Before that, you can go for vector scanning, whether any vector contamination is there, because you are using vector, you are using adapter. So is there any adapter sequence is there? Because that will create a problem. That will be treated as a part of the genome. So we have to remove those 5' and 3' in few sequence we have to clipped off. So trigonometric is another tool. Then we will go for KMR analysis, or we can go for to identify the best KMR, because that will be helpful in case of assembly. And you can use any tool, Abyss, Ray, for genome assembly. Okay, so this is why GBS is very important, because you are reducing the complexity and the size of the data. Now GWAS is also very useful technology. Okay, because sometimes we have a historical population. For example, crop trees. In case of crop trees, we cannot breed them, we cannot cross them, because it will take a lot of time. One scientist will start, and 6-7 generation of scientists will it will take to complete the experiment. So for that historical population, for where random breeding is not possible, for example, human being, you cannot say people to randomly do reproduction with another people. So in that case, GWAS is very useful, and also useful for that, not only for population perspective, but also those who are those characters or traits which are controlled by very small alleles. Okay, there are not major alleles, there are small alleles. For example, quantitative trait. For quantitative trait, GWAS is very excellent tool for identification of quantitative trait locus. Okay, so and GWAS is basically rely on the linkage disequilibrium, basically a correlation structure that exists among the DNA variant and the candidate genome. So linkage disequilibrium is the point to remember here, is based on linkage disequilibrium. Okay, so we have phenotype data, we have genotype data. Okay, so we will put this into a software called structure, and we will go for tassel, and the GWAS will be ready, and there will be a map generation that is called a hap map or haplotype map. Okay, and based on that, we can identify that what is the functional SNP that is responsible for a particular trait. Generally for disease, generally for any kind of quantitative trait, we can identify, we can use this technology. Okay, this is very useful technology. And SNP, why I am putting this in SNP? Because SNP marker is the most robust marker in the genome. So using SNP as a genotype, and using your using them in GWAS will be very fruitful, because you will get a functional allele. You will get a functional allele that is responsible for a particular trait. So that's why in GWAS, SNP plays a very important role. Now, if I say about transcriptomics, so transcriptomics basically deals with mRNA. So mRNA, we have to convert it into cDNA, and then we will add adapter at both the end, and we sequence that adapter just like DNA. And in case of DNA, we are using Illumina. Here we can also use Illumina for cDNA also. So like same, but the problem is we have to normalize the data. We have to normalize that data, because in case of RNA, what is the problem? That is not the problem of DNA. The DNA RNA difference is, in case of RNA, there may be multiple times repeat of a particular read. Okay, some gene is overexpressing, some gene is expressing very less. Okay, so this kind of things is there. So to normalize that data, that we have to go for RPKM value. RPKM means read per million KB. Okay, so read per million KB we have to identify. So now this is related to RNA-seq. Okay, what I am telling is this is related to RNA-seq. I will come RNA-seq later. So for every transcriptomic study, if you are using genomics tool in transcriptomics, that normalization is very important. And why normalization is important? Because there is the expression of the genes are not equal. Sometimes there is overexpression, sometimes they are expressing less. Okay, then we can, after read alignment and mapping, we will get fragments of RNA, then we will align them and we will map them with the reference genome. And we can go multiple studies. We can go for what is the organ specific expression. Suppose you are getting tissue from, you have got your RNA or cDNA from leaf, from root, from shoot. So after getting your alignment result, you will identify that, okay, portion of the portion of the DNA is expressing in leaf or expressing in the root. Okay, and we also identify what are the alternate splicing events. Okay, whether if a RNA has multiple product, then there must be a alternate splicing event. Otherwise, these two have not come. We also can detect a novel transcript. This is the beauty, novel transcript, because you are comparing a control with a treatment. Then if we compare these two, we will get a novel transcript. I will come the technology, DDRT, PCR, SSH, these technologies are there from where we can identify a novel transcript. Then transcriptional fusion events, whether two transcript has joined or not, whether transplicing has happened or not, whether differential gene expression analysis is there or not, whether you can identify non-coding DNA, miRNA, and so on. And finally, we can enrich the data and ultimately form a gene ontology, or we can develop a phylogenetic study. So these are the different technologies for transcriptomics. We can go for microarray. If we have differential gene expression study, we can go for microarray. MPSS, massively parallel signature sequences, ClipSeq, cross-link, immunoprecipitation assay, DNA protein interaction checker to identify the DNA protein interaction. Then we have real-time PCR. Yesterday I also discussed real-time PCR is another excellent tool to identify the differential gene expression, whether it is overexpressing or down expressing in respective control. Then we have CAGE and SAGE for make a unigene, to make a unigene or make a assembly that can represent a whole cell. Okay, in system biology, this is very important. We can go for SAGE and CAGE. We can go for partial sequencing, that is called EST. EST single read one pass sequencing of a single cDNA. Okay, that is a fragment of EST is nothing but a fragment of cDNA you have sequenced, and that is representative of that cDNA. It is not like it is representing cDNA 1, 2, 3. Not. If you are getting a sequence, a part of the sequence of the cDNA of cDNA 1, then EST should be representable of that cDNA. Then we can go for RNA-seq. That is now the most widely used technology of the transcriptomics nowadays. Everyone, because if you have fund, you can go for RNA-seq. RNA-seq is very cheap nowadays. And then northern blotting. If you identify a particular fragment is present in our gene in our pool of RNA, then we have to go for northern blotting. And there are several direct sequencing method also. You can directly sequence the RNA only. No need to convert it into cDNA. From RNA itself, we can do sequencing. So again, after this kind of sequencing technology, we have to go for quality check, assembly, annotation. That same process as in case of genome assembly. First QC, thermometric, and then go for BLAST2GO, and then data analysis. Okay. Now, if we say about the RNA-seq, is the most powerful high throughput transcriptomic tool nowadays. So what we do, we generally in this technology, transcripts are converted into cDNA, and the adapter are ligated as like DNA sequencing platform. What is the required? I told that the library preparation is same for any kind of DNA technology platform, DNA sequencing platform. So you have added the adapter to both the site, ligated the adapter, and the sequencing process of a single read or pair read based on your demand. That is your depth, how much depth you require, how much time you have, how much cost you can bear. Because pair end sequencing, you are sequencing from both sides, 3' and 5'. So cost is more, time is more. So based on your demand, it is there. And millions of short reads will be produced, and they are assembled using de novo or reference assembly. De novo assembly means when you don't have any information of the reference, and if you have reference sequence, then you can go for reference sequence based algorithm. For example, there are so many tools, Bouty, Tophet, Star. Okay, these are very popular tools. Bouty, Tophet, Star, these are very popular tools for sequence alignment. Okay, and then we have to go for quality check, that fast QC, as I told, or quali map. These kind of tools are there. And after that, the remediation of the poor quality read. If you are getting some poor quality read, or you are getting some ligated product, both the sites, adapter sequence, so remove it by trigonometric method. Okay, the fast-text toolkit are there, so you can remediate those poor quality read and then go for NGS data analysis. And after alignment, some tools are there for identification of the quantification of the expression or identification of the transcript, and Scaffolding, CLC Genomics, Selfish. There are few tools, Scaffolding, CLC Genomics. Remember these names and try to read it from any online publications, or you can search, as I told, you can go for Blast2Go tool. You can install the Blast2Go tool and see and check what are the different tools for what purposes are there. Because sometimes the important tool, that is Scaffolding, something maybe it has been asked, why what is the tool for detection of differential gene expression? So Scaffolding is a tool. Okay, and as I told, normalization is very much required because some are highly expressed and some are less expressed. So to normalize the data, we have to go for RBKM value, that is read per kilo base pair per million map read. That is very important. What is RPKV? Read per kilo base per million map read, or FPKM भी बोलते हैं. FPKM, what is FPKM? It's fragment per kilo base per million read. The meaning is same. That means how many reads are there per kilo base per million map read. Okay. Now for differential gene expression pattern, as I told, the CAFTIF, DEXEC, AGR tools are there, and you have to go for fold change expression. How much fold it has increased with respect to control. Okay, then there are tools also. And besides this, RNA-seq is superior in respect to low background noise. Other technology, that is, for example, microarray. Microarray was very useful technology 10, 15 years ago. But the major problem is there is so much background noise. So RNA-seq has minimum background noise. This is the beauty of this technology. Now coming to ChIP-seq. ChIP-seq is generally chromatin immunoprecipitation technology, and this is useful in case of you want to find that what is the protein binding region or transcription factor binding region, or how DNA, how protein and RNA are interacting. Okay, that's why ChIP-seq is another important tool for transcriptomics. So almost we know that almost all the RNA molecule that has some degree of post-transcriptional gene regulation. Okay, for example, capping, polyadenylation, splicing, editing, these kind of things are happening in the with the RNA. So how this process happens, how at what region capping is happening, at what region polyadenylation is happening, where splicing is happening, what are the players of splicing, how gene editing is happening, how RNA is getting degraded, how a RNA is getting transported from a nucleus to the other organelle. So these kind of things we can analyze through chip sequencing, because wherever there is a protein interacting with the RNA, we can go for chip sequencing. So here I mentioned the cross-linked protein with DNA. Actually, chip assay is the name chip suggests chromatin immunoprecipitation. The name chromatin means here we are dealing with DNA. But if we now ChIP-seq is a genomic tool. But if we say clip means cross-linking and immunoprecipitation sequence. The mechanism is same. In ChIP-seq we are using DNA protein interaction, and in ClIP-seq we are using RNA protein interaction. That is the difference, but the principle is same. Okay. So what we did in this process, that RNA or RNA protein complexes are cross-linked. Okay, so you have to cross-link the RNA and protein. First, you have to degrade the total chromosome. Wherever there is a protein is binding with the DNA, they will be intact. And for making it intact, we have to give them UV treatment so that it will cross-link. Okay, this is simple step. Then after diation and immobilization, the antibody couple beat. So there will be antibody that will bind with that particular cross-linked RNA protein assembly. Okay, so there will be an antibody that will be bind to the cross-linking RNA protein complex. Now what we do deep phosphorylation. If we do deep phosphorylation, what happens? So we have captured, based on antibody antigen interaction, we have captured those proteins which are which proteins are binding with the RNA. We want to just identify which part of the RNA is involved for binding of the protein. So after capturing of the based on antigen antibody interaction, we don't need the protein only. So we will remove the protein. For that, dephosphorylation is done. So protein will be removed and the RNA will be remaining. Now we have to sequence this RNA. This is simple. You sequence that RNA and you will get the information of at which position of the protein, which part of the RNA was involved in binding with the protein. So this is very simple. So we can go for mapping thereafter. Now coming to microarray. Microarray is another transcriptomic tool that was very much useful 10-15 years ago, but nowadays also sometimes we have to use it when we have no option for RNA-seq. So DNA microarray, another thing is it can be multiplexed. Okay, and okay, let me tell about the technology first. The potentiality of the technique is high throughput, automated, and we can get a quantitative data also as well as qualitative data also. Okay, so you have a genomic library or Cdna library or EST. EST is another, I told, a part of Cdna. So these are representing of a gene. Okay, and they are basically they placed in a platform, and that each platform representing a particular gene. So you have a platform that, for example, here one platform is showing you have a platform and you have a spot, and that spot is representing a particular Cdna or particular gene. Now you have your sample in your hand. You have isolated your RNA from the sample, and you want to check whether this gene I already have put it into the platform, whether it's present in our sample or not. For that, we will simply do hybridization, and you will get a signal. This is the simple technology based on DNA or nucleic acid hybridization technology. Okay, just remember, in the chip, each probe is located, and each probe means probe means not like a fluorescently labeled DNA fragment. Here probe means a genomic a cDNA, a gene, a complete gene. Okay, remember, here probe means a information of the gene you have put it into the platform. That is a probe. Okay, so each spot has a gene ID. Definitely each spot has a gene ID. So this is the process I told. You have a sample, you have extracted the RNA, so you have converted into series. Now you are preparing an array, that is a platform, that you have a plate, and then you put each gene, a single gene, in a well, and now the array is ready. Now you do the hybridization. After scanning, you will get. Now coming to SSH. SSH is suppressive subtractive hybridization. This is also very useful technology. Okay, SSH, and some noise is there. Is there anybody who can help? Please mute yourself. Now coming to SSH. SSH is a concept that Larrick, Seibert and Larrick in 1992. That was almost 30 years back. And this is also called comparative PCR. Okay, comparative PCR, SSH. So this is an effective method for constructing subtracted DNA library. That means you have a control and you have a treated sample. Then you just want to identify that what novel gene is expressing in the control or in the treatment. Understood? So just a differential gene expression study, not by change fold. Just you want to identify what is the novel gene present in the control or in the treatment. Okay, so for that, SSH is very useful. So it is based on reassociation kinetics. Okay, if we come into this picture, it is based on the reassociation kinetics of the molecules. So the subtracted DNA library can be used effectively as a probe capturing homology sequence or any kind of expressed mRNA data. Okay, so you have control sample. That control sample is considered as driver. Just see the picture. The control sample is considered as a driver. Okay, and the treated sample considered as a test. First mRNA, you have isolated the mRNA of both the samples, control and treatment, convert it into cDNA using reverse transcriptase PCR, and the tester cDNA is then digested using a restriction enzyme. The tester cDNA, tester means your treated sample, is then digested with restriction enzyme to generate a blunt end, and the tester cDNA get divided into two parts. Okay. After digestion with the restriction enzyme, we will divide the tester cDNA into two parts. One part and two part has two different adapters. That's why we gave them digestion to add adapter, and we will we have two kind of adapter, so that we have divided the tester sample in two parts. Okay, and each part has a function. I'm coming. Each part is ligated to one kind of adapter, and after adapter ligation, the tester cDNA is amplified using adapter specific primer. Adapter, we added adapter so that we can amplify the cDNA, so that we can design the adapter specific primer, because we don't know the sequence of cDNA what is present in the treatment. Okay, that's why we have to add adapter. Now the driver tester hybridization will be took place. First we will denature them. As I told, this mechanism is based on reassociation kinetics, so we have to denature them. Now it will get single stranded. Now adapter tester will hybridize. If in what case they will hybridize? If there is a similarity of the RNA expression of cDNA with the control and treatment. If control and treatment, those RNA which are similar in control and treatment will hybridize, and rest of things will not hybridize. Okay, so now you have a one kind of refinement. Those are hybridizing, that means those are common in both the cases. So remove them. We don't need them. We only need the exception one. Okay, the differentially expressed one. Further, the second cycle of PCR takes place, and both the tester having different adapters are mixed and amplified using adapter specific primers. Now this leads to the amplification of the suppress amplification of the common cDNA, because we already removed the common RNAs, common cDNAs. So now what we are getting is only differentially expressed. Okay. The reason behind application of this technology is to quantify the driver cDNA. The driver cDNA which suppress the amplification of the common cDNA present in the driver, and tester to allow the amplification of the novel transcript. So if we identify a novel transcript, SSH is a technology based on reassociation kinetics. You have to remember that. And we are removing the common part of the both the cDNA, and we are only expressing the differentially expressed genes. So this is very beautiful technology. If you have to identify a novel transcript, okay, then DDRT-PCR. DDRT-PCR is differentially DDRT is differential display device transcription PCR, and this is also very useful if you have to identify a transcript that is novel or unique. Okay, so this has a widespread uses. Okay, and first of all, as we can see that we have to isolate the RNA, and then we will go for cDNA synthesis and using oligo-DT primer, and we will pool the sample. We'll pool the sample, and we have to perform the first step of the RT-PCR sample is isolated from the different conditions using a set of oligo-DT primer. So we did the RT-PCR, and we added poly-A tail at the mRNA. Okay, and then we pool the cDNAs. The second step is the PCR amplification of the poly-A tail to generate a pooled cDNA. Now we will convert this RNA into cDNA, and again using the pool sample all. So we have pooled cDNA from all the samples. Clear? Now we will go for a separation in the or electrophoresis. So there will be a separation in the electrophoresis, and the reproducible pattern we will get using in the gel, that is called multiplexing, because you are using so many primers. so many arbitrary primers, because we don't know what is the component of the cDNA. Okay, so while amplifying the cDNA, we are using arbitrary primers. That's why here is mentioned to perform the PCR with arbitrary decamer. Okay, so generally a decamer means 10 nucleotide, or maybe you can go for 20 mar or 25 mar, 26 mar. So this is because we don't know what are the end sequence of the cDNA. So after amplifying, we are using random decamer. Now we have resolved it in the gel, and we will go for sequencing. This is the differential display real-time PCR. And now coming to SAGE. SAGE is serial analysis of gene expression. There is very powerful technology, okay, for gene expression pattern analysis of the cDNA. But here what we do, we have to cleave the sequence. Okay, so in mRNA with poly A tail is converted into cDNA. First, you have to convert it into cDNA using oligo-DT primer. Okay, and then we have to digest it with a frequent cutter. Frequent cutter means that is very rare. We have to the frequent cutter restriction enzyme, generally tetra cutter we use, and then the 3' end of the cDNA will be bind with biotin streptavidin bead. So here is a bead that has a biotin, and here is the cDNA which here is the cDNA which is labeled with biotin, and here is the streptavidin bead when your fragment of the cDNA is attached. So basically this is nothing but like EST. As I told, for EST we are using a single pass cDNA sequence or a fraction of the cDNA that is sequenced. But in case of SAGE, we are using so many RNAs together to convert it into the cDNA, and we are cutting this cDNA and joining it to a bead. Now a linker is added, that is called SAGE tag. This is called SAGE tag. A linker is added, and that is generally 9 to 14 base pair length of a linker, and sequence similarity present in the end will be added, because linker sequence, we know there are so many linkers. As much as linker you possible, you just put it into the solution, and each linker, whenever getting a similarity with the end fragment of the cDNA, will bind. So it will represent that cDNA. As I told, that EST representing a particular cDNA. Similarly, one bead representing one cDNA or one gene in case of SAGE technology. So there will be a lot of fragment that has an independent linker, and if we join these linkers, it will form a unigene. That means a stretch of fragment, or stretch of a fragment that is comprising a sequence of cDNA and a part of linker will representing the whole genome. This is the unigene. This is the beauty of this technology. If you have the SAGE sequence, you can say, I have the whole chromosome, because repository or archiving is an art. Okay, how you are representing a data. You have the whole genome, then you cannot say that I have this much of RNA, that means this much of RNA is expressing. No, RNA, as I told, transcriptome and proteome is a time specific, tissue specific. Okay, so the expression of a gene is time specific and tissue specific. So whenever you are extracting RNA from a sample, you don't need the whole sample. You have to normalize that data. You cleave the data with restriction enzyme. You add a linker, and you put all the linker together. Now you have a stretch of linker containing sequence that is representative of the whole genome that is expressing at a particular time and a particular tissue. This is SAGE. Okay. Now MPSS, Brenner in 2000 gave the concept of MPSS, the massively parallel signature sequence. Is basically a hybrid of microarray technology and a sampling method. Okay, so in this technology, what we have a flow cell that consists of million of DNA that is tagged in a microbead. As we can see, a million microbead is there, and a DNA fragment is tagged. Okay, so the principle behind it is that cDNA clone that are sequenced after a round of cleavage of restriction enzyme. As I said, in case of SAGE, you have a sequence, you have to restrict it, and you have a cleavage of that particular fragment. Now the fragment is small. Okay, then you add an adapter In case of Sage, we add a linker. Now here we add an adapter. Okay, and the adapter can decode the rest of the bases of the overhang left by the restriction enzymes. The adapter is able to decode the rest of the bases that present in the overhang. Okay, so this is the adapter. Now the adapter has a code, the barcode. You can see there's a barcode we already attached. So this barcode is unique for which is attached with that particular fragment. Okay, now you can identify based on the barcode of which gene that I have been tagged. Okay, now decoding is generally achieved by conjugated labels. Okay, then we can analyze it by the cytometry or FACS, fluorescent activated cell sorting method. I will discuss these facts at the last part. So in case of MPSS, what happens? We have a fragment of cDNA that is attached with adapter that already have a barcode, and we can ligate so many adapters together, and we can multiply because we know the adapter sequence. So we can multiply also, and we can repeat the steps for using it as a differential gene expression study. Okay, so this is the beauty of MPSS, and this is very useful because it is multiplexed. So many sample you can do together because you are not dealing with whole length of the cDNA, whole length of the RNA. You are dealing with a small part of the RNA, so this is very useful. Now another important aspect is RACE. RACE is called rapid amplification of cDNAs. What our technology I am discussing is transcriptomics tools. Okay. Now RACE, there are two kinds of RACE, 5' 3' RACE. What RACE? When we have to use? This is important to remember. Rapid amplification of cDNA. As the name suggests, this is very easy to remember that when you have to identify the 5' and 3' sequence, you don't know. You have a sequence information of only intermediate region, but you don't know what is the end sequence. So for that reason, we can go for RACE. Okay, so nothing. This is very simple. That you have the first step is the convert for in case of 5' RACE. That means you want to identify the 5' region of the mRNA. So for that, the conversion of the mRNA to cDNA, then primer that is already known, which is specific at the internal region. Then we can design a primer, and so this is the inter region blue color. So and then we can synthesize the cDNA. That means this part will move to the in this region using RT-PCR, and then a poly A tailing we can add a 3' region. 3' region poly A tailing is very much common. As you know, the poly A can be added at 3' region. So poly A tailing can be done. So poly A polyadenylase, and then denaturation can be done to remove the region, to remove the upper strand, and you can amplify now from using poly T primer. Okay, so just we have added a poly A so that we can get a region from where we can design a forward primer. Now this region is amplified. See, this region is amplified. Now you can sequence it and get to know what is the region. Now in case of 3' also, the similar technology is there for 3' region also. So just this is the mRNA. You know the intermediate sequence. So just you have added few nucleotide adapter has been added at the poly T region after reverse transcription. You have mRNA, so if you do reverse transcription, there will be poly T, and you have added a adapter that has a multiple random sequence. That's why it is mentioned in any sequence you can add at the 5' region of the second strand, newly synthesized strand. Now what we do, we have internal region that is known, and we identify what is this region. So we will design the primer and amplify. Okay, now you will get this sequence poly A with complementary to this region. Now you can do a PCR with adapter specific primer, because this adapter you have designed, you know what is the sequence, random sequence is there, so you can design a primer from that region only. Now you can amplify this product and do sequencing, so you will get to know what is the 3' end region. So this is the beauty of 5' and 3'. RACE, you must remember, RACE is useful when you have to identify the 5' or 3' region that is unknown, and you have a known sequence in between, that then only you can go for RAS. Now coming to proteomics, there are proteomics is the study of protein as a part of functional genomics, as we know, but at a particular time and particular tissue. Okay, so proteomics encompasses the protein mining. Matlab, you are finding something, you are finding some new protein. What is the protein expression profile? What are the network of the protein? How, in which pathway it is involving, and the mapping of the protein. So for every technology, for every aspect, there is a different technology. If you have to identify, you can go for 2D electrophoresis, as I discussed yesterday, the isoelectric focusing and mass spectrometry, isoelectric focusing and SDS page, and we can go for mass spectrometry also. Based on mass by charge ratio, we can separate those proteins. Okay, and if you have to build a network, that means if two proteins are interacting or not, is two hybrid assay, okay, is there? We can go for fast display method also. And if we have to identify the protein sequence, then we have to go for mass spectrometry. Okay, Sanger based protein sequencing method is also there, and mass spectrometry is also there to identify the sequence of the protein. Okay, so the expression proteomics, electrophoresis, protein chips, DNA chips, SAGE can also be done not only for RNA but protein also. Mass spectrometry, microsequencing, these are for expressional proteomics. You have to check the expression. For functional proteomics, that what is the function? So you can go for affinity chromatography, ligand chip based chip assay, chromatin immunoprecipitation, these kind of things you can do. Is type 2 hybrid assay, motif analysis, CD search, concept domain search. These are for functional proteomics. And structural proteomics, you can go for X-ray crystallography, NMR, okay, CD spectrometry. These are for structural prediction. What kind of 3D structure is there? What is the because based on the structure, so many things you can interfere. Okay. So these are the different levels of protein expression, because protein has not only role in expression, because it has a role from starting. Okay, a protein generally stays only for 2 minutes to 10 minutes. Okay, this is the protein lifetime, but in between it has lots of work. It has to do replication as DNA polymerase. It has transcription for RNA polymerase. You can see here, these are all proteins, and these are all doing so many works. So protein study is very difficult, very complicated, and because there is no technology by which you can amplify the protein. So whatever protein you are getting, that much amount is naturally existing. So we have to identify within that arena only. So this is very difficult. So current proteomics technologies, as I already discussed, that 2D spectrometry for protein profiling, separation, purification. Okay, protein identification. For protein identification, you can go for protein mass fingerprinting, electrospray ionization, tandem mass spectrometry, this kind of. And for quantitative proteomics, we can go for ICAT, isotope coated affinity tag. These kind of technologies are there. So 2D gel, I yesterday discussed that you can go for a isoelectric point based separation, and then you can just simply go for vertical gel electrophoresis, that is SDS page. So first of all, based on charge, and then secondly, based on mass, we can separate. Okay, then we can go for MALDI-TOF. What is MALDI-TOF? MALDI-TOF is a mass spectrometry by which we can separate the protein molecules or protein atoms based on their mass by charge ratio. Okay, so what we do, we generally do a tissue separation. We have to isolate the protein, the raw protein, then we will lysate it, we'll digest it. Okay, then we will separate it. Okay, then after separation, we will digest it. So isolate the protein, separate the protein using 2D spectrometry, 2D gel, then we can digest it with protein digestive enzyme. Then we will go for LCMS, liquid chromatography mass spectrometry, combined method we can go for. And then as I told, liquid chromatography yesterday we discussed, and mass spectrometry, as I said, the separation is based on mass by charge, M by Z ratio. So after that, we will check this information or the sequence of the protein we got in the database. So in database, what do we do? As I earlier told, that we can go for alignment. First, we have to align the fragments, whatever the result we got from mass spectrometry, we have to align them, and we have to check in de novo assembly and for comparative assembly also. And we can also check the annotation pattern. What protein it is, what may be the what may be the function. So these kind of things we can do. So this is how mass spectrometry look. It has a source. It has a source. That means you are putting your sample, that is electrospray ionization and MALDI, matrix assisted laser desorption ionization. The name suggests that it has a matrix, and there will be a laser. There will be laser, and that laser will be absorbed by the sample, and it will get simulated, and there will be an ionic movement. Okay, so it will the molecule present in the sample will get excited and will convert into ion with the help of laser. This is the principle of MALDI. So in the source, the ion is formed, and it will go through a pipe that is called mass analyzer. Mass analyzer is basically a sieve which is passing the molecule based on their size, that is mass by charge ratio. And sometimes there are different types of mass analyzer, that is time of flight. So based on time of flight, how much time it takes to move from start point to end point of the pipeline, whatever is moving, or the ion, so that time will be detected. Okay, and then the detection will be takes place. There are so many kind of detectors are there. What is the principle? That is the source, that will be mass analyzer, and mass analyzer generally takes the time of flight, and then detector will be detect the fragment. Okay, so the same thing I already mentioned, and this will be the result. Okay, the result will be like this. So after digestion, the protein may be of different fragment, small fragments. The small fragments, suppose a fragment having only 2 amino acids, a fragment having 3 amino acids, 4 amino acids. So accordingly, just like DNA sequencing, here similar kind of sequencing method is there. So you remember Sanger sequencing, 2 bases, 3 bases, 4 bases. Whenever the DDNTPs was inserted, there will be a fragment termination. Here also, whenever there is a laser, there will be a cut. So you will get different length of peptides, and these peptide length will be measured based on their time of flight measurement. Okay, so this kind of peak you will get, and you have to see here. I have mentioned that now you will get amino acid sequences. Now amino acid sequences will be aligned, and you will search for a database. The database, if the database has anything to say, so based on homology or orthology or paralogy, it will say that okay, this is this kind of protein. Okay, so this is a slide just to compare the few types of mass analyzers, and the good one is TOF. Here we can see for every time it is saying very good, excellent. The sensitivity, speed, okay, in terms of cost, in terms of range and resolving power, everything is very good. So TOF is an excellent mass analyzer tool or you can say for protein mass fingerprinting. So together these things, we can develop a system biology. Okay. Now coming to proteomics tools, Is2Hybrid is one of the most useful technology. Okay, so here we can see that actually to check the interaction of the protein versus protein interaction can be checked through Is2Hybrid, and this is very robust technology and very useful technology, because handling with protein is very difficult. Handling with protein is not easy as like our DNA or RNA. So here what we did, we have suppose we want to check if we have a X protein and a Y protein, and we want to check whether these two proteins have any kind of interaction pattern or not. So for that, we separated. For example, we have a bait and a prey. Just one fragment is called bait, that is a DNA binding domain. Another part is prey, that is that will interact with the bait. Okay, so if we can clone these two fragment cDNA, okay, not protein. Okay, we have to clone that cDNA of X and cDNA of Y in different organism, and we can make a transformation into the yeast That's why the name is2 hybrid. So we can transform the yeast and we can check if there is any change in the marker we have given in the selection media. Okay, so now this is the construct. Okay, now in the understanding will be more convenient for you. For example, you have a lag Z. As we know, lag Z is an excellent marker. Okay, so lag Z is not, this is native lag Z. Okay, when GAL4 is present, this is native lag. GAL4 is a substrate, so this is the native lag Z. Now what we did for GAL4 expression, this protein has two parts, okay, X and Y. Now if we give only X, lag Z is not expressing. Okay, if we give Y protein, lag Z is not expressing. But if we give both, the lag Z is expressing. That means the reported gene indicates, the gene expression indicates that XY, whenever you are giving XY together, they are giving expression. But individually, they are not giving expression. That means X and Y must have an interaction. So this is useful because we need this technology in so many aspects. For example, if a pathogen attacks into a plant, it will release an effector, and there will be interaction of the effector molecule with the different transcription factor present, okay, of a plant to express a particular gene. For example, yesterday I told talent. Talent is the effector molecule. It is interacting with different transcription factors, and it will helps in genome editing. So there are in different aspects, because transcription factors, no transcription factors alone do a single work. For example, splicing. In case of splicing, that is a housekeeping technology, housekeeping process. In splicing also, you can see there are so many players that they form a spliceosome complex. So many players are there who can interact with each of them and give a output. So is2 hybrid is very useful for study the protein-protein interaction. Now coming to comparative genomics. Comparative genomics is two terminology you have to remember. I'm not going into more details. The synteny and collinearity. What is the? For example, you have an ancestral species, that is the reference genome ABC. Now you have species A and species B. For example, rice and wheat. It is told that rice and wheat were divergent from a evolution was based on divergence followed by duplication of a particular different chromosomes. That means once upon a time, rice and wheat were same, were from the same origin. Now they are belonging to different, they are now to different species. Okay, so not only species, they are now different genus. So what is the reason? The reason is because by evolution there may be any kind of divergence, duplications of the genome due to the pressure of the selection, due to the pressure of the environment, and so many reasons. Geographical parameters are also there. So there is the changes of the genome sequences, genome order, etc., etc. So what is synteny and what is collinearity? For example, the ancestor has ABC. Now they have diverted into two species. Species A has ABC, A dash B dash C dash, or species B has A double dash B double dash C double dash. There may be a condition that all the genes are somehow changed. A is not similar to A dash, but somehow similar. Okay. Or there may be a condition that not only sequence has been changed, but the order also has been changed. Okay. See, is ABC, now it became ACB, now, or in species CBA. So there are two conditions: whether gene order and sequence both are changed, or whether only sequence has changed. If the sequence has changed, now if we compare these two things, when the sequence is the only problem but order is the same, that means that is collinearity. So in case of collinearity, the genome location, gene location, genome sequence, and genome order. If we are comparing both the aspects from order and sequence, then this is collinearity. And if you are only comparing based on the physical colocalization, okay, then this is called synteny. Okay, just remember what is synteny and what is collinearity. And there are different tools for forward and reverse genetics. What is forward genetics? When you have phenotype, you want to identify the gene sequence, that is forward genetics, and vice versa. If you have the genome sequence, but you have to identify the phenotype, that is reverse genetics. Okay, so you can go for different trapping methods. Okay, different trapping methods. That means you can go for genome enhancer trap, promoter trap, gene trap. There are so many trapping methods available. So what are these things? Basically, if you see the picture very carefully, you have given the boxes. Boxes means exon. Okay, boxes means exon, the red color, and the lines are introns. These are introns. Lines are introns. Now, enhancer trap construct the minimal promoter of a reporter gene, that is a TATA. Okay, minimal promoter means the promoter which is regulating the gene of expression is activated by an enhancer. Whether this promoter is activated by enhancer is not, for that we have inserted a minimal promoter. We have constructed, this is our engineered product. So we have constructed a minimal promoter of a reporter, and if with a reporter gene, okay, this is the reporter gene. If this gene is controlled by an enhancer, then definitely this enhancer will also act as an enhancer is a cis element that is helping a promoter to express their genes. First, to understand what is enhancer. Enhancer is a cis element that is present in the same chromosome of the gene, but from the distal region, it is controlling the expression of a gene. So if I have to identify whether this gene is controlled by enhancer or not, we will just upstream of the gene, we will put a minimal promoter along with a reporter gene. If this reporter gene is expressing, that means enhancer is controlling the gene of interest. Okay. Now, in case of promoter trap, what we do, the promoterless reporter gene can be expressed when inserted, occurs in the exon of the such transcription fusion. So what happens? We have, this was the gene, we have inserted a reporter gene in between, which we do not have any promoter. In case of enhancer trap, there is a promoter because we want to check the enhancer is working or not. But here our case is whether promoter is working or not. For that, what happened? We have inserted a reporter gene in between the gene. So whenever the gene will express, there will be a fusion product of the gene as well as the promoter. There will be translational fusion, transcriptional fusion. Okay, the promoter is trapped now. In case of gene trap, the promoterless reporter gene contains a splicing acceptor site. Okay, and expression of the reporter gene occurs when insert into an intron. So in between an intron, I have put it a reporter gene which has a splicing acceptor site. Okay, you also know, you must know there is a splicing donor and splicing acceptor site in between the exons. So at the 5' end of the first exon, there is a donor site, and the 3' end of the second exon, there is this acceptor site. So there is a trans-esterification reaction happens when splicing happens. So if there is a splicing happening and the gene is expressing, that means gene has been trapped. Okay, yes, this is a gene, you can say. So this is, and tilling is another important technology. I did not find any good picture, but whatever the message from this technology is, you have to remember the full form: targeted induced local lesions in the genome is a method of is an efficient technique for mutagenesis. Fine. You have to identify a mutation. For example, you have a population, you have given EMS means point mutation, and you have a normal. So this is the normal and this is mutated. You just denature it, control, and treat it. One is treated with EMS, one is non-treated with EMS. So you denature it and reassociate it. So there will be a mismatch whenever there is a SNP. Because of EMS, there will be a SNP. So there is a mismatch, and you just digest with cell 1 and do electrophoresis, or you can go for HPLC also. Then you will identify which portion of the genome there is a SNP if you sequence the genome. Other tools are there. FACS, as I already discussed, FACS, where when you have a particular cell with a particular reporter gene expressing, and you want to identify which kind of gene it is that, so you have to capture the cell only. Or suppose you want to sequence it, or you want to get a transcript result of a single cell. So identify a single cell is very difficult, and for that FACS is there, fluorescence activated cell sorting. Okay, Bonner identified this technology in 1972. Here we can separate based on ion or based on fluorescence. or based on different wavelength of laser treatment, you can whatever the desired desirability of your experiment, so you can change the detector method. Okay, so this is basically whenever you want to capture a single cell, you have to go for FACS. Then you have to identify methylation pattern. So for methylation pattern, there is methyl sensitive enzyme out there, for example MSP1. There is a methylation, there will be a cut just upstream of the methylation. Okay, so if we get that kind of fragment from the whole genome, you cut it using MSP, there will be small different fragments, and those fragments are representing of the methylation site of the particular genome, and you can align it with the reference genome. You will get to know where is the methylation is happening in the genome. You can go for affinity maturation also. Just remember the name for identification of methylation. So one is methylation specific enzymes, sensitive enzymes. One can go for methylation maturation, also called MEDIP or MEDIP chip. So here what we do, we have to fragment those genome and then denature it. When wherever methylation is there, there will be a specific antibody specific to the methylation. Then we trap it and fluorescently label it and see the chip. See the chip, whenever there is a methylation, there will be red color. If there is a partial methylation, there will be yellow color, and unmethylated, there will be green. So this kind of technology can be used to identify that where is the methylation and how many methylation is there, whether it is partially methylated or unmethylated. Now the best technology is bisulfite sequencing. You have to identify which portion of the genome or which portion of a fragment of a genome is having a methylation. So for that bisulfite sequencing, what is the basic thing you have to remember? After bisulfite treatment, the unmethylated cytosine converted to thymine. For example, C, if there is methylation, after bisulfite treatment also the C remains C. If there is a methylation, after bisulfite treatment also C remains C. But if there is a non-methylated cytosine, then it will convert into thymine or uracil. Okay, so this is the thing you have to remember. If there is a methylation in the cytosine, it will remain cytosine. If there is not methylated, then after bisulfite treatment, it will convert into T. So this simple thing, after sequencing, you will get to know wherever there is a uracil, that means that was actually cytosine. But wherever there is a cytosine in the present sequencing technology, that means that C is methylated. So bisulfite sequencing is very useful. Then DNA footprinting, again, to identify the protein binding region of a DNA. Simply, you can cut the DNase 1. DNase 1 gives a double-stranded cut randomly in the genome, randomly. Okay, so DNase 1 footprinting assay is that you can cut the DNA randomly, but whenever there is a binding of the protein, there will be no cut. So this is your treatment, this is your control. Here protein is binding, here protein is not binding. Keep DNase 1 cut in both the cases. Now, if you compare with lane A and lane B, this is the fragment, this is the fragment. You can compare that there is no fragment in this region. That means no fragment means because there is a protein binding site. So you can say that this is the region where any protein or transcription factor or anything that is binding with the DNA. So you can track that region. So thank you. That is what about genomics and proteomics and transcriptomics. I think most of the things I have covered. Just remember those things I have told, and please read a book, because bioinformatics is a area where you have to remember so many things. Beyond understanding, also you have to remember most of the things and practice it using I told Galaxy, or you can go for Blast2Go. These tools, these are freely available tools. You can use it for practicing using any random data sample taken from NCBI raw data. Thank you. So if anybody having any questions. Okay, I will share list of books. Okay, for each of the topics, for which topic you have to read which book, and any if you are facing any difficulty in bioinformatics, just go for any basic book. Okay, you can go for any tutorials also. There are so many videos are available in the YouTube, so that you can, if don't you for exam point of view, you need not have to remember the mechanism or the principle or the algorithm as such. Okay, because the blast algorithm is also very difficult, but you have to know the what is the blast, what are the different types of blast, what is the parameters of blast. For example, p value, what is p value, what is e value, what is blossom. Okay, these kind of questions are asked in plant science perspective. Okay, because you are not giving exam in bioinformatics, you are giving exam in plant science. So the what is e value, whether high e value is good or low e value is good. Okay, what is blossom, what is different types of blast. These kind of questions will be asked. Okay, and try to remember these things. And I definitely I will suggest a few books, but if you go through the books, don't go very deep into the subject. Okay, just go at a superficial level, what are the names, and try to remember those things. Make a chart and revise it every day. And or else what I told that blast2go and blast2go tool and Galaxy, these are freely available cloud-based tools in the website, so you can check, you can make an account and you can check, and you can just practice with random data set. Okay, so I think how much distance between two? Actually, distance between two gene. Okay, the distance between two gene nowadays. Earlier, it was based on the marker and the gene distance. Okay, so there are different mapping techniques. Okay, mapping techniques is another aspect. QTL mapping, linkage mapping. There are so many mapping technologies, and based on the marker information. Okay, the Bayesian approach. There is a mixed linear model, multiple linear model. So many models are there based on which you can do QTL mapping. And QTL map, whatever you are getting information from the QTL mapping is based on, if you are using genetic mapping data, then you will get what I mean, if you are getting linkage mapping or QTL mapping based on genetic marker data, then it is based on centimorgan. Okay, so centimorgan is a relative term. In case of Arabidopsis, if you compare rice genome, distance between rice in centimorgan would differ from the Arabidopsis. Okay, Arabidopsis, it may be 1 centimorgan equal to 100 base pair. Because today we have the physical mapping, the fine mapping, we know the base pair. So what is the need of centimorgan? So the distance between two genes is nowadays we measure through base pair. Okay, so that's how we do, and that's all. Okay, thank you. I am leaving now, and if you have any query, you can mail. I am giving my mail ID. So you can mail me, and if you need the slide, I can also share with you. I will send it to Dr. Bhale Rao sir, and he will give it to you. Okay. And the slides itself is not sufficient. Try to read books. Slide is only for representative form. Make the slide as your theme and read it in depth from any book. Okay. So thank you very much. All the best for your exams, and thank you. I'm leaving. Okay.
Thank you, sir.